Welcome, folks, back to microbiology. Today we're going to be talking about chapter 6, which is microbial nutrients and growth. Now, we had kind of prefaced this discussion with our previous chapter, chapter 5, where we talked about why we need nutrients in the first place. And that is because of the energy that's being used in the cell to drive metabolic processes. So the energy that's being extracted from the nutrients that's coming into the cells is being used to make energy transfers, using the energy from the chemical bonds to extract, store, and transfer energy. And these involve oftentimes the movement of electrons and molecular components like ATP to transfer energy between energy states, and then couple those transfers of energy with both anabolic and catabolic reactions. So our cells can continuously move to be organized, or the cells of bacteria can continuously move to be organized and maintain their organized complexity in a world that is naturally moving towards entropy or disorganization. And so that's really the preface for why we need nutrients. And today we're going to be talking about some of those nutrients, not just nutrients, but also physical requirements for growth that microorganisms need. So I would recommend pausing the video here, reviewing some of the previous content, and coming back in. Okay, welcome back. So for today's lecture, we're basically going to be talking about three kind of chunks of information and then tying it together at the end. So it'd be really useful for you to take notes in kind of three big overarching diagrams or concept maps or sections, for example. So the first big section that we'll talk about are what are the nutrient and growth requirements for organisms. Okay, so that's one part, and you can kind of divide up into the different types of nutrients or the different types of requirements that we discuss and the different types of example organisms, for example. The second big part is how we then utilize those requirements that we know organisms need to be able to grow them in the lab. Okay, so we're going to talk specifically about media and different types of media, a lot of which we will use in the lab, specifically in our kind of in-person hands-on labs. And then the third section is where we talk about growth phases of microorganisms and bacteria, and kind of how they move through their life and their growth as a population. And so that's specifically going to focus on a diagram like figure 6.20. And so you could like annotate that diagram and have an idea of that section. So those three big concepts could really translate really well into three big diagrams or three big note sections. So just a recommendation there. So let's jump in. So our discussion today is essentially, what do microorganisms need to be happy, right? And so for microorganisms to grow and then reproduce, and when we say growth in this chapter, oftentimes we're actually talking about reproduction because microorganisms kind of have this basic structure or shape and size, and they don't really kind of grow and grow and grow and maximize on that size necessarily. They kind of maintain a specific size, but they do grow in number exponentially oftentimes. And so that's kind of our discussion for today. If you have optimal requirements for growth, then you will have oftentimes exponential growth rates in that specific colony or population. Okay. So a couple of terms when we're talking about growth in the lab setting, microbial growth in the lab setting, oftentimes we'll refer to an individual colony, and that is a group of organisms that have come from one individual original colony forming unit, and they grow on a flat media in the lab. Whereas a biofilm is kind of a complex mix of community of microorganisms, either in the lab setting or in the environment, medical setting, so on and so forth. And so essentially that is what's going to result when microorganisms are in an optimal setting, either in the lab or in a more community-based natural state. Okay. So here is an example of colonies. We're going to look more we're going to see this played again later on, and then I'll give you a link to how we actually do this method where we can see individual colonies, or we might see microorganisms in various settings in the environment where they actually grow in aggregate. And we're going to talk about how they might do this and why that might be beneficial to them. Okay. So when we're talking about nutrients, some of the really core essential nutrients for microbial growth are carbon, oxygen, nitrogen, and hydrogen. And we're going to kind of go through some of these. We're also going to talk about electrons and kind of where those sources are for microorganisms. And that gives us an idea then of that second section that I talked to you about how we can then grow them in the lab rather than just interacting with them in the complex environments that they live in, right? So when we're talking about, let's start with carbon, energy, and electrons. So where are organisms getting their carbon from? Where are they getting their energy source from? And where are they getting their electrons from? And we have some terms that allow us to kind of distinguish between these sources, okay? And they'll have a table that summarizes these terms as well. So when we're talking about carbon, you could either have organisms that use an inorganic carbon source, specifically carbon dioxide, and these are called autotrophs because they're able to kind of incorporate on their own from an inorganic source that carbon, rather than heterotrophs, which have to utilize some existing carbon source for their some existing, molecular organic molecule for their carbon source. When we're talking about energy, the primary source of energy for communities and ecosystems is the sun. So you have a group of organisms that are called phototrophs, and these are organisms that are directly able to utilize solar energy. And then, like us, there are many organisms, even in the microbial world, that are not able to use solar energy, but then rely on the products of phototrophs and other organic molecules within their settings to then actually harvest energy, organic and inorganic molecules. And so those are chemotrophs. Okay. You do have this diagram that breaks some of that down, but I do want to add another set of terms to this diagram, and I'll show you an updated diagram as well. So electrons are also a really essential kind of component, as we talked about in the last chapter, of chemical reactions. And so you have organisms that are able to utilize electrons from organic molecules or organotrophs. And then you have organisms that are more self-sufficient, and they don't need an organic source for their electrons. They can actually just use inorganic sources, like many photosynthetic organisms that are using, for example, water as their electron source. And these are called lithotrophs, litho referring to inorganic molecules. And so we actually name groups of organisms in this way. So phototroph, chemotroph, autotroph, lithotroph. That gives us an idea of where they're getting these various components from: their energy, their carbon, and their electrons. And so this table breaks those down. And so you can see the energy source, either sunlight or chemical energy, carbon source, either inorganic carbon, like specifically CO2, or organic carbon sources, and electrons being from inorganic sources. So we use the prefix litho, or organic sources, organo. And so we can break that down. These are full words, but I've just put the spaces in them so we can see what they mean. So many plants and photosynthetic organisms are photolithoautotrophs. So they are getting their energy from the sun and they're getting inorganic molecules as their electron source. So water, for example, and they're getting inorganic molecules, specifically carbon dioxide, as their carbon source. Okay, and troph refers to energy. So overall, this is how kind of they're getting their energy and what they need for chemical reactions. Whereas many heterotrophic organisms like ourselves, we cannot use solar energy. So we need either inorganic or organic chemicals coming in. And this actually specifies that we specifically need organic chemicals for our energy source and for our electrons as well. So those oftentimes it's the same nutrients that we take in or the food that we take in that we get both of these from, the energy and our electrons. And hetero means that we're not able to use inorganic carbon. We need organic carbon. So heterotrophs. Okay, and so just like in the macro world where we have plants autotrophs, and heterotrophs like us, in the micro world, we have the same diversity and complexity of microorganisms, where you have some organisms that are photosynthetic, some organisms that are living in deep sea vents, so they don't have access to sunlight, for example, but they do have access to chemicals that they can use. Or you have organisms that are actually heterotrophic, and they're eating up organic molecules, for example, to extract their nutrient requirements, electron and energy requirements, okay? And so anything that's autotrophic tends to be more self-sufficient than organisms that are heterotrophic. This becomes really important when talking about something like a pathogen. It's less likely that you'll find an autotroph that's a pathogen because they don't really need organic molecules for their living systems and their metabolic processes, whereas many pathogens are heterotrophs because they're utilizing our body in some way to extract the organic material that they need for their metabolic functions, right? And so these distinctions become really essential. Then there are a lot of other components, either nutrients or different types of, environmental components or situations that are really essential for understanding how an organism is going to exist and, thrive in their environment. And one of these is oxygen. So let me just move to that section here. Okay. So oxygen is really essential to microorganisms specifically, and organisms generally, because oxygen has some very unique characteristics. And so we saw in chapter 5 how oxygen is a really optimal electron acceptor, and that's great, but sometimes it accepts electrons off of molecules that it shouldn't be accepting electrons off of, essentially steals electrons from those molecules, which can inhibit metabolic processes. So oxygen is kind of a double-edged sword within the living world, within the biological world. And so you have some organisms, especially at the microbial level, that can utilize oxygen because it is so optimal at being an electron acceptor within cellular respiration, for example. And you have organisms that cannot utilize oxygen. And then you have organisms in between that are like, if it's present, it doesn't hurt me, but I'm not going to actually utilize it. And so we had a little bit of a discussion about that in chapter 5. And we can continue that discussion here, where essentially oxygen in a healthy kind of metabolic process or chemical reaction is just taking electrons off of, for example, electron transport chain, right? But it can actually turn into a free radical where it takes electrons off of molecules that it shouldn't. And we can kind of see that here in this video. Okay. And the reason why it does that is because oxygen in its atomic form has 2 unpaired valence electrons. And so those 2 unpaired valence electrons are constantly looking for electrons to interact with and pair up with. And so if it finds a protein or another molecule just within the cell that has electrons that are easy to remove, it will do that. And that starts to cause cell damage and cell aging. And so within the cell, we have a number of enzymes and a number of different molecules that help act as antioxidants to protect our cells from that kind of damage. And so they can donate electrons to the oxygen in a way that doesn't kind of inhibit metabolic processes, or they can deactivate that oxygen by attaching it to another molecule or something like that. And so in terms of, let's get to this slide here, in terms of the different types of microorganisms that kind of, and the spectrum of how they interact with that oxygen, you have organisms that do have those enzymes, specifically any organism that exists within an aerobic setting and utilizes oxygen needs to have superoxide dismutase, which is an enzyme, and various types of catalases. And so these, what we call oxidases and catalases, allow for it to protect itself against the damage of oxygen and also utilize oxygen. Then you have microorganisms that just have the enzymes that they need to detoxify the oxygen, but not to actually utilize oxygen. So they only have some of a subcategory of these enzymes. And so they can tolerate oxygen, but not actually utilize it. And that gives us a spectrum from strict aerobes to strict anaerobes and about everything in between, okay? And we'll see this really clearly in the lab. And the way we'll see this is we'll inoculate these, what we call deeps, that have very specific chemicals in them that allow us to see where the oxygen is in the tube and where the growth is in the tube. And so if we see that growth correlates with the presence of oxygen, then we can say pretty clearly that that's an obligate aerobe. It's an organism that has all of the enzymes it needs to protect itself and utilize oxygen. Whereas if we only see growth at the very bottom of the tube where there's no oxygen, then that's an obligate anaerobe. Now we also have in our lab facultative anaerobes. Facultative anaerobes, for me, it's kind of a misnomer because these are actually aerobic organisms that can facultatively exist within an anaerobic environment. And so they prefer oxygen, but if there's no oxygen, they can actually switch to fermentation, or they can find other ways of utilizing that environment and those nutrients. And so they're facultatively anaerobic. And then we also have aerotolerant anaerobes, which are a possibility. They exist in the environment. We don't work with them in the lab, but this is what we would see if we had them cultured in the lab, where they would grow through and through the tube. They are anaerobic organisms. They don't use oxygen, but they do have the enzymes they need to detoxify their cells and their environment from the harmful impacts of free radicals. And so they are able to grow through and through the tube. Now, we don't work with aerotolerant anaerobes, but they do exist in the environment. Okay. There are also some organisms that are called microaerophiles. Microaerophiles are able to use a little bit of oxygen. They have just a small amount of those enzymes that they need to utilize oxygen. And these specifically are kind of in the groups of Helicobacter and Campylobacter, which are Helicobacter you might recognize as being important for ulcers, for example, where they can utilize just a little bit of oxygen, about 2 to 10% of oxygen. We also don't work with these in the lab, but if we did, we would see them grow in these tubes just below where the highest concentrations of oxygen are, and that very high concentration of oxygen at the top, we wouldn't see so much growth. And so those are those kind of two specific categories of gram-negative organisms that are called microaerophiles. Okay. Another really important nutrient for growth is nitrogen. Nitrogen is essential for many macromolecules that we utilize in our cell construction. And so it's really important to be able to access nitrogen. We talked about nitrogen a little bit. It's kind of funny because it's prolific in the environment, but very difficult to access. And so you have a number of bacteria that are able to fix nitrogen and then release the nitrogen in a useful form for the rest of the organisms in the community, which is very beautiful. Although we have found artificial processes and methods to do this, which we have talked about a little bit previously in this class. We've also looked at some of those microorganisms in the lab. So for example, these microorganisms that are in the nodules of these pea plants, which we interact with in our lab and we actually grow in our lab. Okay. There are some other nutrients that are needed like phosphorus and sulfur and other what we call trace elements that are needed in small amounts as cofactors, for example, for enzymes that are important to microbial growth generally. And so we need to be thoughtful that these are in the media that we're using in the labs for us to actually be able to grow these microorganisms. Okay, so that gives us an idea of kind of the requirements for nutrients or various types of molecules and elements that a microorganism might need. Now let's talk about the kind of physical environment that it's living in and some of the requirements that it might need for growth. Okay, so some really essential ones are going to be your temperature, pH, salinity, so on and so forth. We'll kind of take those in turn. So in terms of temperature, with living systems generally, and microorganisms as well, as you increase the temperature, you increase the kinetic motion of molecules and enzymes within a cell. And that allows them to interact with each other more frequently and for reactions to actually happen more frequently or more quickly and more smoothly. But there's a threshold. And when you get to a certain kinetic level of kinetic energy, those molecules and enzymes start to break down. And so generally, as you increase the temperature, you tend to get an increase in bacterial growth and activity to a point at a threshold which those microorganisms and their chemical processes are not able to continue. Okay. Now, that optimal temperature and that threshold depend on the microorganism and where they've existed and evolved in the world or what communities they exist in. So you have extremophiles that live in very hot, or in this case, very cold temperatures. This is a green algae, but it actually makes a red pigment that kind of covers up that green color. Photosynthetic algae that lives on glaciers and snowfields in the Alps and polar regions. It's called Chlamydomonas nivalis. And so it's very interesting that it can kind of live in these extreme environments, and it's able to kind of utilize that very, kind of lots of solar energy that comes into a glacial area like that, even though the temperatures are very cold. There's just a fun optional video about this microorganism in the description below that you're welcome to watch. Okay. Another really important essential environmental characteristic is going to be pH. So is the pH optimal for that specific microorganism? You have many microorganisms that are existing within about a neutral pH. A lot of the pathogens of a human are going to exist within a neutral pH unless they're pathogens of the digestive tract, which you might have acidophiles in that area. But you do have organisms that are able to utilize very acidic or very alkaline environments, like various, hot springs and things like that. And so those organisms, whatever pH that organism exists in, that pH is the one at which their enzymes and their proteins tend to be stable at. If you change the pH to too acidic or too alkaline outside of that range, then their enzymes and proteins start to break down and they're no longer able to exist in that environment. So a macro example is ceviche, which is a food item that we essentially cook the proteins in just using an acid, right? And so that acid destabilizes the proteins in that shrimp and gives it the cooked texture, right? And so making sure that microorganisms are living in a pH that's optimal to them. Okay. Water requirements are essential to microorganisms. Every cell, whether it's part of a macroorganism or it's a microorganism, a multicellular organism or it's a unicellular microorganism, is essentially just a bag of water, right? With a membrane that's semi-permeable, but usually permeable to water. And so being really aware of water requirements. So you have organisms that are able to exist through desiccation, for example. Maybe they produce endospores or something like that, and many organisms that aren't. And also tied to water is salinity, because whenever you change the salinity in the outside environment, then you're going to impact the flow of water in and out of that microorganisms. So an example from the macro world, that we know not to drink salt water, right? Because that actually dehydrates our digestive system and our cells. Same for microorganisms, unless that microorganism is halotolerant. We will actually work with some microorganisms in the lab that are halotolerant, and you'll see some media that we use that actually selects microorganisms out by adding salt to the media. We'll see that in today's lecture. But unless those organisms are halotolerant or able to tolerate high levels of salt, or halophiles, actually salt loving, then they tend not to exist in really salty environments. Okay. Now, when we grow microorganisms in the lab, we want to know exactly what are the nutrient requirements and what are the environmental requirements for the growth of that microorganism. Okay? And we tend to try to recreate those in the lab setting. And so in the next part, we're going to talk about the media that we use in the lab and how we select that media to be able to grow specific types of microorganisms. But before we get into that very detailed discussion, I want to remind us that microorganisms like us do not live in isolated kind of petri dishes in isolation. They live in biofilms or communities, what we call biofilms, where other microorganisms are together and they utilize different parts or different nutrients within that environment or that setting, and they tend to work together. And these biofilms exist naturally in the environment, and we can kind of imagine them as just as we imagine a macro world, right? And so if you'll step into the macro world for a minute with me and imagine that you're flying on a plane and you'll look down at night and you'll see city centers where there's lights. And that's because where there are resources, there are people and there are ecosystems. And where there are not resources, you won't see as large of an aggregation of the human population or, within a natural system, an ecosystem. And so it's the same with microorganisms, the same concept. You'll see them aggregating around resources in the natural world or in the built world. And they actually have mechanisms for seeking each other out. When they have what's called quorum sensing, and quorum sensing is when microorganisms can actually detect the metabolites of other microorganisms. And when they find those are higher in concentration, they will move towards those metabolites. They will move towards the aggregate of other microorganisms, because that is an indication that there are nutrients in that area that they could also utilize along with the other microorganisms in that biofilm, right? And so this is what happens in the natural world. You have an aggregate of microorganisms, and they actually intentionally aggregate by seeking each other out through quorum sensing. And so we deal with this on a daily basis: the plaque on our teeth, biofilms on medical equipment, biofilms on various types of industrial equipment or marine surfaces, or biofilms within the home setting, right? And so this is how microorganisms exist naturally. When we grow them in the lab, we separate them out. and we use specific media for specific microorganisms, and we try to isolate them to learn more about who they are and their characteristics, which is not really the natural setting, but it does give us a lot of really important information. And that's one of the reasons why you culture microorganisms in the lab on petri dishes, but you also have these rhizobia that you grow in the garden by planting peas. So you can kind of get that perspective that microorganisms are living in community in natural settings, right? Not just in these isolated petri dishes. So with that kind of little bit of perspective, we're going to move into the next part of chapter 6 and talk about how we actually manipulate media in the lab so that we can grow specific types of microorganisms. And then we'll talk about microbial growth and the phases of microbial growth. So thanks for tuning in so far. You can jump into the next part and we'll continue this discussion.